So welcome back, everyone, for the third talk of day 3 of the workshop. It's also a talk which is a colloquium for C3.ai DTI Center. It's our great pleasure to host Vince Poor. So Vince Poor is the Michael Henry Strater University professor at Princeton, where he's engaged in research in information theory, machine learning, and network science, and their applications to wireless networks, energy systems, and related fields, including recently modeling COVID-19 epidemic. He's a member of National Academy of Engineering and National Academy of Sciences. He's a foreign member of Chinese Academy of Sciences and the Royal Society. Recognition of his work includes a 2017 Alexander Graham Bell Medal and honorary doctorate from universities in Asia, Europe, and North America. Personally, I first came to know Vince when he was on sabbatical at Stanford in 2000, if I remember correctly. And since then, of course, he's a great role model to follow. So with that, Vince, I'm going to mute myself and remove myself from video to avoid attention or distraction, so to speak.
Let me get my screen cleaned up. Okay, great. Well, thank you, Devavrat, for that kind introduction. And thank you and your co-organizers for inviting me to speak this afternoon. I'm really happy to be doing that, and also very happy to be speaking in the UIUC colloquium series as well. So today, the title of my talk is fairly explanatory. I'm going to talk about how to incorporate evolutionary adaptations into spreading models over complex networks. And I'm really going to focus mainly on epidemic modeling. And in fact, as was indicated in my the brief biography, we're interested really, of course, in looking at improving the models of how COVID-19 has spread. That said, I'm not an epidemiologist or a virologist, so my perspective is going to be one that I think is familiar to most of the audience here, which is from the point of view of network science and electrical engineering and so forth. But the work that I'm going to talk about is the beginnings of several research projects that we have going on at Princeton, funded by some of the what I would call rapid funding instruments that came out shortly after COVID emerged in the United States. There's an ARO grant, an NSF rapid grant, and very relevant to this workshop, a C3.ai DTI grant, which is joint with several people whose names you see on the screen, particularly Osman Yegan at Carnegie Mellon, with whom I've been working on a lot of these problems for a while. Simon Levin, who's a mathematical biologist at Princeton, Josh Plotkin, who's a mathematical biologist at Penn, Rashad Eletreby, who is a former PhD student of Osman's, who's now at Walmart Labs up in Jersey City, and then a postdoc, Ben Schaffer, working with Simon and me, and a grad student, Ani Sridhar, working with Osman and me. So this is a team effort. There are some biologists involved, and we're just now starting to develop some models that are relevant to the COVID spread. What I'm going to talk about today are just some basic ideas behind that. The way we got interested in this problem, Osman and myself, was that we were interested through another ARO grant in the problems of information spread in complex networks. And one of the problems we were interested in was how adaptations of messages change that spread. And of course, it's a very similar problem, as everyone here knows, I'm sure, to the spread of pathogens in an epidemic. And so we got interested also in how adaptations could affect the spread of pathogens through a complex network of human connections. We wrote a paper on that back last fall. It appeared in PNAS in March, right at the time when the COVID epidemic was really taking off. So we got the idea that perhaps we might be able to use some of those ideas in modeling COVID. So I'm going to talk about some very basic ideas. I'm not going to use any real data at all here. Part of what we're doing now is trying to map real data onto the models. But that's basically the genesis of this, and this is what I'm going to be talking about today. So let me, there we go. So first of all, what are spreading processes? Or, well, you know, we're interested in questions about like the following: what causes a disease to spread widely and become an epidemic? And obviously not all diseases become epidemics. So what causes that? Or what makes information go viral? And of course, spreading models are models that help illuminate these phenomena. So that's basically why we're interested in spreading models. And today, as I said at the beginning, I'm going to focus not on all spreading models, but just on epidemic models with mutations. And the epidemic modeling goes way back. There are many, many different types of epidemic models. So really the thing that we're mostly focusing on here are the effects of mutations. So I'm going to begin with perhaps some, for many of you, an unnecessary background on epidemic modeling very briefly. And then I want to talk about a little bit about motivating the study of multiple strains in such models. And then I'll talk about some simulations, a way of modeling mutations, adding mutations to some standard spreading models, and look at some very basic simulations with just very synthetic, just to give you an idea of how the parameters of the model come into play. And then I'm going to talk about an intervention, a particular intervention that's, of course, in the news a lot today, which is mask wearing. And it turns out that we can use a model of multiple strains and mutation to model mask wearing. That is, we can think wearing a mask or not wearing a mask as being two different mutations or two different strains in a spreading model. So we'll see how we might use these modeling techniques for looking at interventions. And then I'll wrap up. So this is all very basic, but at the end, I'll talk about some open issues that I think will make these models more relevant to what's happening today. Okay, so let me just talk very briefly about modeling of epidemics. So, of course, the goals of, and we're talking about modeling, of course, in this community, we're talking about mathematical modeling. And the goals of mathematical modeling of epidemics is just to predict their spread. Okay, so the spread of something like COVID. And also then, once we have a good model, we can sort of use it to understand the impact of various countermeasures like mask wearing, quarantines, contact tracing, and so forth. And I think any of you who've read the news or keep up with the news over the past year have seen how varied the models have been and how varied the predictions have been of the models that are out there. And so there's still a lot of room, even though epidemic modeling is an old field, there's still a lot of room for improvements, I think, and we've seen that in the current pandemic. So we're going to look at agent-based models. I'll say a bit more about this in a second, but the basic idea is you have some initial patient who's infected, and then the pathogen spreads from person to person through a contact network. So this is a so-called agent-based model. So more generally, spreading models of multiple kinds sort of start out with a basic model called the SIR model, which I'm sure many of you are familiar with, if not from your work, from the news, where again, you have one patient who's initially infected, all the others are susceptible. And then as people come into contact with the infected patients, they may become infected, and then they may also infect others. Eventually they may recover, or they become not susceptible. They could also die, as of course, unfortunately happens a lot with COVID. So that's SIR is the basic kind of model. There are variants. There's SI, where you don't recover, and HIV, for example. Although now people do recover from HIV, but that was for a while was an SI type model. SIRS, where you can recover but then become susceptible again. And we're seeing with COVID that in fact seems to be a possibility. There's SEIR, where E is exposed, so you can be exposed and pass on the virus without being infected. And we've seen that, and that's another feature of COVID. And then other variations on those themes. There's quite a bit of work in this area. So within this SIR modeling, there's two basic types. There are population models where you track the fraction of individuals of each of the types of population. And you describe that through a differential equation. And you just assume basically that any two individuals can interact with one another. And so this doesn't really take into account the contact network. Unlike agent-based models, which I mentioned before, where the idea is we put the network, there's a network topology involved. And so we're basically looking how the pathogen spreads through a contact network. So the network has a role here, unlike in the other model. Okay. And so although this is, of course, when you have differential equations, it's much easier to think about mathematically. In agent-based models, there are asymptotics and so forth that allow you to make analytical predictions. So I'm going to be looking at those kind of models here. So let me motivate a little bit now the specific type of models we're going to look at, which are those that consider multiple strains and mutations. Basically, my understanding is a virus is really just a nucleic acid wrapped in some kind of protein. So it's very, very primitive type of organism or molecule, and it can mutate very easily. So in fact, most of the pathogens that have spread or become infectious diseases in modern times have actually mutated from animals. So they're zoonotic. I think there have been 400, as I understand it, since 1940 infectious diseases, and I'm not sure whether those are new ones or just total, but 60% of those apparently came from animals. So in coming from animals, they mutate to be more transmissible from human to human, and also maybe to be more virulent in certain ways. So as viruses are spreading through the population, they mutate. So that's one of the things to consider. And the mutations of the virus may have different properties from the original virus. Okay. So this is something that's not typically accounted for in the models that exist, and that's what I'm going to work on today. Also, although I'm going to talk mainly about epidemics here today, the same thing happens, of course, in information spread. Almost 10 years ago, we had the beginnings of the Arab Spring. There was a lot of social media around that. A lot of the news spread that way. And there was a lot of misinformation, a lot of things that spread differently, and people viewing the same event reported it in many different ways. And of course, even without that, we know that information spreads from person to person, it evolves as it goes through. If you think about Twitter, if a tweet becomes a little more provocative, it may spread more rapidly than if it's just bland. So you can see that information, just like pathogens, can spread, can evolve, and that evolution may affect their spread. And of course, this particular pandemic, the SARS-CoV-2, which is the pathogen that causes the COVID-19 disease, has in fact mutated. And originally they thought the mutations were not important, but more recently there have been a lot of news stories about the importance of some of the mutations. So it's particularly relevant to the current pandemic and perhaps to future ones as well. So for example, there are thousands actually of mutations of the SARS-CoV-2 virus, and different ones have been prevalent in different parts of the world. And here's a map, I think. Yeah, there's a map here just to show that different parts of the world are affected by different strains of the same virus, basically. So what does that mean for modeling of the virus? I mentioned this work with Rashad and Osman and a few other co-authors, which appeared in PNAS. And basically, in that work, we studied agent-based models with multiple strains and mutations. So I'm going to talk a little bit more about that as we go forward. But it allowed us to develop analytic predictions for certain things, probability that epidemic emerges, and the fraction of infected individuals, that sort of thing. And we also saw from that work that if you ignore mutations, you're going to get incorrect predictions when mutations exist. And I'll give some examples to show that later. So here's a very simple way to model this. So in the SIR model, you start out with an infected patient, and that infected patient has a certain probability of infecting each of its neighbors. And that probability, in this case, we'll call it T1, is called the transmissibility. Now, you might have heard of R0, which is the average number of people infected by an infected person. And that would be something like T1 times the average out degree of the graph, the connected graph of all these people. Okay, so T1 is the probability of transmissibility. Now, in the SIR model, a certain fraction of these people would be, these neighbors would be infected. They would also have a transmissibility of T1, and this process would go forward in that way. But in our model, after people are infected, there's a possibility that the pathogen might mutate within that person before it's passed on. So, for example, there may be two strains, strain 1 and strain 2, and after being infected, some of the infected hosts may, the virus may mutate within some of the affected hosts before it's passed on from them. So we can think about a matrix of conditional probabilities. So mu11 is the probability that mutation 1 stays as mutation 1. Mu12 is the probability that mutation 1 changes to mutation 2 or in a new host, and so forth. So if we have M possible strains, then there's an M by M matrix of probabilities, which just transition probabilities from one strain to the other, evolving in a given host. For example, if we have two strains, once neighbors of the initial host are infected, there's some probability with which the strain will remain as strain 1, and there's another probability with which the strain will mutate into strain 2 before the next iteration of the spread. And then for strain 2, there's a separate transmissibility, T2. So we have T1 and T2, and from that host, there'll be a certain probability T2 of infecting neighbors. Within those neighbors, they may maintain strain 2, or could mutate back to strain 1 or to some other strain. In this case, is only 2, or it could stay the same, and so forth. So I think everyone sees this. You just propagate forward, and until basically the model stops when there are no more people that are susceptible, that are connected to infected hosts. Okay. So basically in this model, I'll just go back. So if we have M possible strains, and then each one has a transmissibility T sub 1 through T sub M. Well, the product of a diagonal matrix with those T's on it times U, if you look at the spectral radius of that times the average out degree of the nodes, you'll get what's the R0 for a model like this. Okay. Okay, so in that PNAS paper, basically we studied this model analytically and through simulation using some networks from real world, schools, hospitals, Facebook, Twitter, and so forth. And basically the things you can do, as you will see if you look at this paper, the things you'll do is you can predict the epidemic threshold, that is the parameter levels at which an epidemic starts to spread, the expected epidemic size, and the expected fraction of individuals infected by each strain in a multi-strain model. Okay, so this allows us to look at how all these things interact with one another, which you can see is useful for thinking about not only how modeling the spread, but also perhaps modeling how changing the parameters by modifying the graph through some restrictions or mask wearing or what have you might control a spread. And the other bottom line of this paper is basically that if you don't consider multiple strains when they are there, you'll underestimate typically the virulence of a pathogen in forming an epidemic. So let me illustrate that through some examples. So here's just a simple baseline example where we have a single strain. So this is not a multi-strain model, and the transmissibility is 0.3. So this is just to get you show you what the charts look like. And here, time here is dimensionless, so it's not there's not a particular time of interest here. But in fact, you can see on the left, you see versus time, the fraction of nodes infected. You can see there's a peak here around 3 time units, then eventually dies down. And on the right, you can see basically the cumulative number of infected nodes. And this is for a simulation of 5,000 nodes, 5,000 node networks with a Poisson degree distribution with mean 10, averaged over 50 trials. I see now, by the way, that I've gone about 20 minutes, so maybe I'll stop here before I move on further. Deborah, any questions out there?
Yes, there's this Madhav here, Vince. Thanks.
Hi.
Hi. So you described how the mutated virus might go to two hosts, the green ones. What happens in the SIR model, we also need to figure out if you and me infect Devrath, for instance, and you carry one of the strains and I carry one, what's the tie-breaking rule in your case? So you toss a coin, whichever one infects, you keep that host effectively, or that strain. Is that the model?
Yeah, that's right. Mm-hmm. Yeah. Good question. Okay. Yeah. Other questions? Okay, I think I'm just going to go on. Okay, good. So just again, this is a baseline example, single strain SIR model, classic, and just to get you to see what the charts look like. On the left, we're showing instantaneous number of nodes versus time. On the right, cumulative number of nodes versus time. So now let's look at a 2-strain case where strain 1 has a very small probability of mutating to a highly contagious strain. So strain 1 is the strain we just looked at. Strain 2 is a strain that has a transmissibility of 0.9. So it's very contagious. But the probability of mutating is very, very small. So there are 3 cases shown here. One is no mutation from strain 1 to strain 2. One is mutation with probability 0.001, and the other is mutation with probability 0.003. And then those 3 cases are shown in blue, orange, and green, respectively, in the charts below. So blue is exactly what we showed before. There's no mutation, so it's just a single strain model. You can see that as we allow larger probability, albeit quite small, of strain 2, the percentage of the nodes infected at any time, or the peak anyway, goes up, and the peak moves earlier in time. And if you look over at the right, you can see that when we have any small probability of going to that more contagious strain, in the end, basically everyone is infected. Whereas before, there was a small fraction of people who were not infected. Okay, so just a very tiny probability of mutation causes basically full infection of the whole population. And this really just shows the number of strain 1, the fraction basically of nodes that are infected with strain 1 for the model I just showed you, with the 3 different possible probabilities of mutating to strain 2. You can see that the blue is the same as the baseline case. As we allow a small probability of going to strain 2 or a slightly larger probability of going to strain 2, strain 1 is basically suppressed in the population, and strain 1 and strain 2 takes over. I think I might have yeah, so what this shows is, so this is strain 1, okay, and this shows the same thing for strain 2. Of course, with the blue, there's no strain 2, so there's no fraction of strain 2 there. But once strain 2 is allowed to get into the system, it really takes over and infects most of the population. So, it makes sense. This is very highly contagious, so it's going to take over and get there before strain 1 does. We can also look at other types of modeling situations. We can look at a situation which I understand from epidemiologists often happens, is that viruses mutate to less significant strains. And this is an example like that. So here you have 5 strains, and you can only go from one strain to the next. You can't go back. And each strain is increasingly less transmissible. And so here you can see on the left the fraction of nodes that get infected by each strain in this model, and the total number of nodes infected by each strain. So in the end, this last strain is first of all not very transmissible, and secondly, it's only you have to take 5 stages of mutation to get to it. So you can see why it wouldn't be very prevalent in the population. So that's a very simple look at this multi-strain model. And now I want to also look at how we might use it to analyze interventions such as mask wearing. Okay. So we can think of mask wearing as being 2 strains of the same disease. Someone wears a mask, they have one transmissibility. If someone doesn't wear a mask, they have another transmissibility. Right. So we can think about, let's forget about multiple strains of the virus now and just assume there's a single strain of the virus propagating through the network. But some people wear masks and some people don't. So those are really the 2 different strains, if you will, of the model. So we can think about transmissibilities depending on whether the host and the target are both wearing masks, one of them is wearing a mask, or both not wearing masks. And so we have 4 numbers: TMM, which is the probability of transmission between 2 mask-wearing individuals; TMN, which is the probability of a mask-wearing individual infecting a non-mask-wearing one; and similarly, TNN and TNM, so going from a non-mask-wearing individual to a non-mask-wearing individual, or a non-mask-wearing individual to a masked individual. Okay. So those are reasonable parameters to think about. So how might we use the model I just discussed to look at the effects of mask wearing in such a model? Well, first of all, we need some kind of innate or prior idea of what fraction of the population are going to be wearing masks. But so for example, let's let P be the probability with which an individual wears a mask, and just assume it's independent from individual to individual, although that of course may not be true. But let's just assume it is for now. We can change that also in the model. So then we could think about a 2-strain model with 2 transmissibilities, one for mask wearers and one for non-mask wearers. So for example, if for a mask wearer, the probability of a mask wearer transmitting to another mask wearer is basically the probability of the transmissibility in that case times the probability that a target is a mask wearer, plus the other transmissibility to a non-mask wearer times that probability. So we can set up basically 2 transmissibilities, one for mask wearers and one for non-mask wearers as the hosts. So similarly, the non-mask wearing, there's a similar mixture of the 2 probabilities. We can also set up transition probabilities from mask wearing to non-mask wearing. So we can think about going from mask wearing, say, to non-mask wearing in the target as being a mutation. And we know all the conditional probabilities of that. So we can use Bayes' formula to get the probability if the initial node is a mask wearer and a node is infected, we can write the probability that the affected node is a non-mask wearer using Bayes' formula. And this is how you do it. The bottom is just the total transmissibility, the top is the transmissibility to a non-mask wearer. So similarly, we can look at the other transition probability as well. So we can take all this and just set up a model, which is really an approximate model, but it should be pretty good for a large network, with two transmissibilities and transitions from mask wearing to non-mask wearing strains and vice versa. And then we can just use the model we looked at to make predictions for the mask wearing model. So if we can set up a simulation here, suppose that the transmissibility of mask wearing to mask wearing is 0.2, mask wearing to non-mask wearing is 0.3, non-mask wearing to mask wearing is 0.4, in keeping with the idea that it's more to protect others than yourself. And then non to non to 0.6. So these are just numbers we pulled out of the hat, but I think they're reasonably thoughtful numbers for mask wearing certain circumstances. And then let's look at the probability from 0 to 1 in increments of a quarter, okay, of the population that wear masks. So in this case, the blue is no one wears a mask, and of course, this is now transmissibility is 0.2, so it's not quite the baseline here, but it's very close, very similar. Goes up to a little over 50% fairly quickly. As you start having higher and higher fraction of people a population wearing masks, the fraction of infected population goes down a lot, and the peak is pushed out, which is something, by the way, that epidemiologists have told us about interventions. Is one thing it does is it pushes the peak out, which is important for resources and so forth. So this is a toy example, but it illustrates that property as well. And then you can see on the right that if everyone wears a mask, according to this model, it's really going to keep the epidemic under control. We can also look at that was using what I was just showing you, was using the two-strain model. If we actually run the mask wearing model, it's slightly different until you get a very large network. And if we look at the asymptotic value of the fraction of infected individuals versus the fraction of people wearing a mask, you can see the circles are using the mask model and the squares are using the mutation model, which I just showed you. And in that paper I mentioned, there's an analytical prediction of the mutation model, and you can see that the simulation matches it almost perfectly there. Okay, so I'm a little early, but I'm going to go ahead and wrap up, and we can take time for questions. So here's what I've talked about today. First of all, I think the most important thing is that mutation is an important factor in the spread of an epidemic. And what we've tried to do here is to get at some simple way of introducing that into standard models. And I mentioned this work of Eletreby and all, where we there are analytical predictions. I didn't talk much about them here other than just to give examples. And I didn't show examples, but in that paper, you can see examples where models without mutations give incorrect results. And then I also talked about using that same model to model mask wearing as a two-strain propagation model. So I want to mention a few open issues now that I think might come out of this work or continuation of this work. So first of all, one is, all the examples I gave were just simple examples with chosen parameters, okay, toy examples. I think the next thing we'd like to do is to look at this model and test it against what's happened with COVID-19 by extracting parameters from data. And C3.ai, of course, has the data lake with many different kinds of data sets that we can do that with. And Ben Schaffer, who I mentioned at the beginning, postdoc, is starting to look at that data and starting to extract those parameters. So maybe in the next talk, I'll be able to talk about some specific things having to do with COVID. Another thing we might look at here is the modeling of other interventions like school and business closings, contact tracing, and quarantines. I mean, these are things that would not affect necessarily the transmissibility directly, except that they might affect the the contact network, right? So if you close the schools or businesses, you start affecting the contact network, which is the point of those closures. Another thing to look at is to introduce multiple strains into the modeling of intervention. So when I looked at that simple mask model, the underlying viral model was just a single strain. But we know that, of course, the virus has multiple strains. So sort of a very easy thing to look at next would be to look at how, for example, how does mask wearing work when you've got multiple strains? I mentioned Simon Levin and Josh Plotkin at the beginning. They're mathematical biologists. They study optimal control of epidemics, mainly through timing of interventions. And so one of the outcomes we hope of this model is that once we have a good model for COVID, that they will be able to understand better when are the best times to enact controls, meaning various types of interventions, and when is the best time to release those controls. My understanding is in other epidemics they've studied, they've shown that optimal controls actually apply interventions twice, and apply them once, let them loose, and then apply them again, and that's the optimal way to control the epidemic. So we'll see if something like that works with these multiple strain models. I think the way Osman and I got interested in this business was through multi-layer networks. So our other ARO grant is really about multi-layer networks. And here, there's some very natural multi-layer networks. In particular, if you look at epidemic spread, and we've seen a lot of this in connection with COVID, there are many different networks that we're involved in: schools, neighborhoods, families, workplaces, and so forth, shopping. And so if you don't count those as a single network, but look at that as multiple layers of a network, then it's easier to see how shutting down schools, for example, might affect the spread, because you can just take out a complete layer of the network by shutting down the schools, and then you can see how things spread otherwise. So that's one of the things that we're also planning to do in this work. And then finally, another thing is to look at non-binary transmissibilities. So in a lot of the models, it's basically transmissibility is binary. You've got, with certain probability, is you transmit the disease or don't transmit it with a complementary probability. But we know from COVID and probably other diseases as well, that it's not a simple picture like that. There's a super spreader. So some people spread the disease much more readily than others. They're asymptomatic people, so they may have a bigger contact network, or they may be spreading it without knowledge of it. The different populations have different responses to COVID. So there are a lot of differences from person to person. So the simple model of you're infected and you have a certain probability of infecting someone else is probably not very accurate for something like COVID, but rather some kind of probability distribution of spread is probably a better way to look at that. So anyway, those are some things that are on our agenda, and hopefully over the next year or so, we'll be talking more about these. But with that, I think I'm done, and that's my second 20 minutes, and there's time for questions. Thank you.
Vince, thank you for an excellent talk. I think I can see that various participants are opening up cameras, asking questions, so I'll let them ask.
Okay, great.
So is there an order to doing this, or do we just speak up?
Yeah, go for it. You're one of the people here, Simon.
Hi, so this is Simon Levin. Vince, great talk. Thanks very much. I probably could keep these comments for when you come home, but you stimulated a couple of thoughts. One is that you really have two contagious processes going on here. One of them, the disease itself, and the other is mask wearing, which is a contagious process. And one of the things we're finding, both from the data and from some models, is that the latter has multiple stable states. If you compare, for example, the Scandinavian countries to the Asian countries, you know that there are huge differences in the number of people who are wearing masks. it's a social norm that can suddenly flip. So a natural next step, it seems to me, is going to be to put both of those processes in at the same time, allowing the spread of the virus and the evolution, perhaps between multiple strains there, and flipping between individuals on these networks and how they might flip categories, susceptible and mask wearing, et cetera, the sorts of things you hinted at.
Yeah, well, that's a good comment. Yeah, I don't think it requires an answer, but it's a great comment. And since we're working together on this, we should get on that right away. Thanks, Simon.
Okay. Stefano, would you like to read out your question that you posted in chat?
Sure. What I wrote was that you spoke about how mutations are likely to be important, but I'm interested to know if you have some insight into how you think they might be important. So, influenza mutates to escape immunity, whether vaccine acquired or naturally acquired. Highly lethal viruses tend to become less lethal because that conveys a selective advantage for them. Viruses can mutate to increase transmissibility to the extent that that conveys a selective advantage, but this is already a pretty transmissible virus. I'm curious as to what you see as being the important aspects of mutation that we need to pay attention to in modeling, but also potentially in response.
That's a great question. That's one of the things that we want to address here. I don't really know. I would suspect that transmissibility is a big one. But looking at that map I showed earlier, you can see there are multiple strains that seem to dominate certain parts of the world, but I don't think we've seen a lot of difference in terms of the way it's affected populations. So they may be all equally virulent, but with different transmissibilities, or just there may be some random luck. I don't know. Simon and I had a conversation about this with Josh not long ago. I don't know if he has any comment he'd like to make about that. But there has been some recent papers that have been circulated on this question. I don't know, Simon, did you have anything?
Sure. We've been interested in this, and in particular, what the impacts of interventions might be. What is social distancing doing to the evolution of the virus? The trade-off is implicit, or maybe even explicit, in the question you put in the chat, which is, indeed, there are advantages to becoming more transmissible, but also advantages to keeping your host alive longer. And so ultimately, you expect some resolution to that trade-off, and that's certainly true for influenza. Some of the early mutations for COVID-19 have been ones which increase transmissibility, largely involving the spike protein. But there, as far as I know, there hasn't been any indication that these are more virulent strains. And it may be, like Vince says, that there's not a great difference in virulence, although often these are coupled because increasing virulence means building up a higher virus titer in the host, which also gets more of it transmitted. So it's hard. So these are ultimately coupled. We really don't know, but it's a great question.
Well, and just taking the HIV example, it doesn't surprise me that different strains are dominating different parts of the world. But in the case of HIV, that doesn't seem to be well correlated with either virulence or transmissibility. And so I wouldn't necessarily expect that here either.
Yeah, well, you would expect HIV, you might think of it as a very virulent type, but there's not much selection for reduced virulence there. I would think because the hosts are staying alive longer and longer. And so there's lots of, transmissibility, as Vince showed, would be the transmission per unit time multiplied times the amount of time that you're transmissible. And people with HIV are transmissible potentially for long periods, untreated for long periods of time.
And in this particular case, most of the mortality occurs after the period of transmissibility, and therefore isn't foreshortening the transmissibility period. So there'll be very little selection pressure to make it less lethal, unlike a disease.
I agree, that's basically what I was saying. I agree with that.
Good.
Yeah. and even with mortality rates of case mortality rates of, I don't know, 1 to 3% or whatever it is for COVID, that's probably not a strong pressure for reduced virulence either.
Okay, I'm glad Simon is on this.
Well, so am I. I get to hear what we're doing.
This is Madhav. We have another question.
Other questions, yeah.
Yes.
Madhav's one of our collaborators as well, of course.
I have a question, and this is another one that Simon and I have started looking at. Do you have insights on the structure of the network and its effect? I probably missed some aspects of it, but did you try out different network structures to understand how that impacts?
Well, these are random networks. I mean, in the paper that I mentioned, there are real networks, hospital networks, school networks, and you get various different results for various types of networks. Yes, so you can. I mean, there are also Facebook and Twitter and so forth and so on. So what we haven't looked at much is different types of random networks, for example. But I think that also would be something interesting. So it's easy enough to do by simulation anyway, not necessarily by analytical result.
Right.
Yeah, I have a question.
Yes.
Yeah, so have you looked into the environmental factors as well for the spread of disease? As in, it's been told that when the temperature affects the transmissibility rate as well.
Well, that's a good question. So at the end, I mentioned this idea about the transmissibility not being a simple binary property, right? So I think that might be incorporated into the transmissibility in some way, turning it in, having other parameters of transmissibility other than just a single probability.
Okay.
We haven't done that, but I mean, the specific answer to your question is no, but I think that the way to do that is pretty clear. I think you could have a richer type of transmissibility, just like you could consider different types of spreading and so forth. But that would be another way to do that.
Doesn't appear that the virus has been damped down in the warmer climates, unfortunately.
Okay.
Yeah. Vince, you might mention the work we're doing with the Metropolitan Opera Orchestra there.
Yeah, so that's really interesting. So this was actually Simon initiated this. The Metropolitan Opera Orchestra is very eager to learn how they can safely start performing again, and they approached Simon about this. And so we're working now with some colleagues at Princeton, particularly Howard Stone, to model how the droplet spread changes from the speaking or breathing to playing instruments and singing opera. The opera singers have very high, number of dB of power, sonic power, and so the idea being to look at how we might modify some of the rules of distancing, for example, and how for example, the orchestra might be placed in different ways to mitigate spread or allow safe performance both by singers and by musicians. I mean, some of them is kind of obvious. I mean, if you're playing a string instrument, you can wear a mask, but if you're playing the flute, you're blowing across the flute. So the way orchestras are designed now, the strings are in the front and the winds are in the back. But it might make more sense, not acoustically, but certainly.
Okay.
And that's not just theory. Howard's doing experiments.
Yeah, we have one opera singer and a couple of musicians coming into the lab.
Yeah, two opera. Isabel Leonard, I think, has been down already. So.
So that should be some kind of coming out soon. There's some. I didn't follow it recently, but I saw some email about there's going to be some press on it.
Yeah, it'll be in the New York Times pretty soon.
There are a few questions that folks from YouTube chat have posted. Vince, I'll just read them out one by one. So first question is from Trevor Miles. The question says that shouldn't we consider the difference, I believe different effects or differences in effects of wearing a mask as a transmitter versus as a receiver?
Yes, and in fact, that was in that model. I maybe went over it a little quickly, but there are different, the transmissibility is different if you're from mask to non-mask as it is from non-mask to mask. And that reflects what we understand from public health experts, that you're really protecting other people when you're wearing a mask more than you're protecting yourself. So that's a good question, but I think it is in the model. And perhaps if you go back and look at the presentation again on the recording, you'll see that aspect. Good question.
Another question is from Ismail Seth. In Mexico, the authorities did not recommend the use of mouth covers because it gives the false sense of security that people do not use it correctly. Can this factor be considered as a part of your model?
Yes, in fact, one of the things that Simon has talked about, and I'll let him speak for himself as well, is that we not only need to do optimal control, but we need to do robust control, robust to factors like that, the fact that people don't actually behave perfectly the way they should. And that can be modeled, right? I think this is as long as you can think about putting some probability on that, I think you can model that as well. Yeah.
Okay, and I think Scott, if you want to read out your question, I presume you're on Zoom.
Yeah, I was wondering if mutation could affect detectability, which would in turn affect the effectiveness of test-based interventions.
Yeah, I'll give that to Simon, because that's more of a biology question. I don't know that I know.
Well, the answer is definitely yes. And in fact, we have a paper where the first author is my student Shadi Saad Roy, which appeared in PNAS some months ago, which was actually before we knew anything about COVID-19, which is that it would be advantageous for a virus to develop an asymptomatic stage, because then it could hide out. First of all, not only not be detected by public health, but not be detected by the immune system. And so that's another sort of trade-off. But it could, a mutation, we've already seen a mutation to develop these asymptomatic stages, and obviously that affects detectability, and that's been a huge problem in terms of the public health treatment. So one can imagine other mutations that would, any mutation that's going to affect symptoms is going to affect detectability, which could fool some of the tests. I think ultimately we need serological tests that are going to detect whether we have antibodies, although there are a lot of suggestions that the people who are recovering are doing so not with the antigen-specific antibodies, but with T-cell responses, which are providing some long-term protections. So the short answer to your question is yes, but I didn't give you the short answer.
Yeah, well, another way, I saw Tamara Bashar is on this call, and it made me think of game theory. You can model that using game theory, right? A game between the tester and the virus for the guy. The virus obviously wants to maximize its spread, and the tester wants to minimize it.
I'll give you a story on that, which I think is amazing. First of all, we had some working on influenza. We had some work which showed that there's something called antibody interference, and if your immune system attaches to sites on the virus, that may block its ability to attach to other sites. If it attaches to a low neutralization site, that could interfere with its ability to attach to the high neutralization site. Well, our collaborator, Peter Nara, pointed out that the virus has actually figured this out and puts out decoys. This is the influenza virus to trick the immune system into attaching to these no neutralization sites, and then it comes back with these with these mutations that can't be blocked. So it's very much a game theoretic problem, and we're often losing.
Insidious.
Yeah. He calls it deceptive imprinting. And we published one paper on that about how that ought to affect vaccine design.
So,
Anything else?
I have a quick question. This is Srikanth. So this is a broad modeling question. Anybody can take this. So I guess over the course of the colloquiums this year and during this workshop, we've seen two types of models. One sort of a branching process type probability model for modeling the disease spread, and the other one is a differential equation model. So what do, when people make decisions, public policy decisions, is it, to me, from a distance, not knowing enough about the area, probabilistic models perhaps are more accurate in the early stages of the epidemic spread, and probably always accurate, but then become maybe intractable after a while, and then you switch to a differential equation model? Or is there some other way to think about the two models and their relationship?
Well, I'll answer a different question. I gave a similar talk at Berkeley a couple of weeks ago, and Shankar Sastry asked whether it might be possible to put mutation, into the differential equation model, which I think is an interesting, another problem. I should have mentioned it in the last slide, but of course it's a continuum model, so you would think that at some point, I don't know whether it's a continuum in density or in the number of nodes, probably in density of the nodes. At some point, they should converge in some way, but I don't know how that convergence works. Some others here who've worked more directly on those models may know, but I would suspect that the differential equations at some point would be as accurate.
Well, I would think the differential equation models are going to be more accurate, are going to be sufficiently accurate for large populations, and when things are fairly well spread. But at the mutational stage, it's a stochastic process, and so it's hard to capture that with the differential equation model. Mutants arise and die out. But,
so in the early stages, branching process models are better.
Yeah, I think that's right. One also has to remember that what early stage means. Even if a particular strain is widespread locally, it might be rare. And so if mutations arise locally, and a stochastic process is probably going to be needed for that stage, even when the virus is pretty widespread.
Srikanth and Madhav here, if I could just add, I don't know whether you meant from branching process network style models or not. You don't necessarily.
Well, both. Network-based branching process or without networks, depending on how you believe people interact. Yeah.
Right, and so I can speak to a little bit of the network versus differential equation-based models, and each of them has its strength, in my opinion. But there are certainly certain questions the network models seem to be more tuned to attack in some form, but it has got other challenges in terms of computation and such. But we find, for instance, school opening is a question we are studying right now. And very simple branching process style model is a better one because it's very hard to capture some of the parameters for understanding the growth of the epidemic in the early process. But I think the difference between the differential equation network itself is very, very interesting. And when one model does better or not is a question. There's a paper that hopefully we'll write up with Simon sometime. It tries to articulate the difference in a particular setting.
Interject here with a small point. We played with some models exactly on this, the branching type of processes or networks and the relationship with differential equations. We have a paper on the bioarchive is waiting for the scientific reports. And what we found is that depending on the network structure, you get ODEs with fractional exponents. And this is quite reliable and robust. So for example, and we tested it against some COVID-19 data. So for example, and it makes sense that the mixing is not perfect, is localized sort of nuclei around the population. And it's at the interface between small areas that the disease propagates. So the mixing, which is the product of S times I, assumes a complete mixing. Everybody talks to everybody else. So if there is some sort of a neighborhood that is accounting for the propagation, then it makes sense that it's not product, but it is a little bit less. And we found something like 0.6, 0.68 is a very good exponent for the I.
Can you share that?
Sure, I can post it on chat, or
yeah, if you post it on chat before the meeting ends and people can copy it, I think it'll survive.
Right. Okay.
Yeah. There are a family of models in the infectious disease literature that use dependence on a power, and we've also done it in a different context. Derived. So I wonder if you can get to some of those limits analytically using what amounts to renormalization approaches.
Yes, the renormalization approach usually assumes it has a strong assumptions, because you have some sort of a degree distribution.
Yeah.
And when you look at possible neighbors over the whole network, you end up having more or less a tree structure. You don't have cycles, triangles, which is not the case, because, you know, if I'm sick and my neighbor is sick, most likely, you know.
Yeah.
You know, so this is sort of the point.
Well, you might also get to it with a kind of pair approximation.
A kind of what?
Pair approximation.
Pair approximation. Yes.
So,
but anyway, if you could share that, then I can share.
Yes. I'm going to do this.
So I know we're at the top of the hour, and of course people are, but in interest of the question that Shrikant asked, maybe sort of let me ask another question. New York at its peak, if you look at it, sort of the number of infections were happening, okay, and you look at that as a fraction of population, and now then project it down to MIT's population, that number would turn out to be something less than 8. Now, if you have one person infected within MIT, likelihood of she or he infecting 8 people in even in sort of a constrained setting is very, very much likely. Which means that if New York's peak was scary and hence one should shut down, that means that sort of university population needs to be shut down. Where am I going with this? That means that any sort of simple, in this small data question world, which is very hard to sort of have data-driven policies. And so we're left with commonsensical policies. And so the question to us here, who are at least the folks who like numbers and equations, what would be collective thoughts on sort of how does one sort of put these two views together?
That's deep.
So Devrat, just to understand your question, you're saying that if you project, so you're talking about a R0 of 8 in case of MIT population, if you project New York numbers, is that what?
I'm not even going to sort of the asymptotics or averages. All I'm saying is that if we believe, okay, so here's a question. What is it really scary number of people being infected in MIT population? I don't know that number. So one way to sort of look at such a benchmark is to say, well, we know what was scary. Definitely what was in the peak in New York, potentially most of us, if not all of us, would agree that that was a scary situation. And if that was a scary situation, I can scale down the number. And maybe that's not right, but let me for a second scale down the number. Then when I would do back of envelope calculation, that number would turn out to be close to 8. That is, I've got 8 infected people in MIT population, and that's a scary number. So, and getting 8 infected people is not too difficult if you have one infected student who actually interacts even reasonably with some other peers. Or maybe
you're talking about an R0 of 8.
Sure.
Yeah. That's definitely scary.
Again, so I mean, I mean, there's an R node and there is a sort of actually contact, right? I mean, there is a sort of,
Yeah,
I think he's saying something different, Simon. He's saying that if if I can rephrase, he's saying if you look at total number of cases in New York, let that be X, and look at the population P, then he's looking that at ratio of X by P, we felt scared about the whole situation. If he looks at that ratio and maps it into MIT population, correct me, David, then that number happens to be 8.
Correct.
At 8, you should be as scared as. Is that a question? Is that a correct understanding?
Right. And then as we collectively, all universities and all local populations think about our own local policies, this question naturally comes up. And so I'm pretty sure all of you have thought about this. And so I thought I'll bring it up. It's, if nothing else, it's a good way for me as a moderator to lead us towards the end of the colloquium. But I'm more actually hoping that some of you would have reactions, maybe not immediately, but maybe tomorrow or in the other breaks as we sort of go through this workshop. Again, so first of all, virtually, Vince, the claps for a very interesting talk and stimulating a very nice discussion here. For all the workshop participants, we'll be back again tomorrow at 1 p.m. Eastern, 10 a.m. Pacific, and all the other time zones in the middle. And we have 3 exciting talks again tomorrow, and Lei, my co-organizer, would be leading the charter of moderating them. So please do plan to attend them, and in the meantime, have a lovely evening.